Inside the brain, these ventricles are known, well, they are actually hollow structures. What does that mean? They're empty spaces. But these spaces are filled with a specific liquid or fluid that is known as cerebral spinal fluid, that expands all the way from the brain to your spine. And what are the structures of your ventricles? You have your laterals, you have your third ventricle, you have your fourth ventricle. Those are your ventricles. Now, what structures allow for the fluid to communicate or circulate actually across between the brain and the spinal cord and back? That is your interventricular foramen. So from the lateral into the third, from the third to the fourth, that is mesencephalic aqueduct or aqueduct of Sylvius or cerebral aqueduct. That's how your lab teaches you, cerebral aqueduct. Okay? And then lastly, out of your fourth ventricle, you have your what? Central canal, which is the one that you find on your spinal cord segment in the middle. You guys remember this kind of drawing, right? So this is your central canal. Okay? That spans all the way out. So, what structure will separate both lateral ventricles? And actually, you saw this on the lab as we dissected. And this is known as the Septum Pellucidum. You guys remember that one? It was a thin sheet. Some of the time, like I cut it through. It was a really thin sheet that separates both of them. Okay. So how do we make this circulation take place? Well, we know there are three meninges in your spinal cord that expands all the way to your brain, right? And those were the what? Pia mater, arachnoid, and dura mater, right? From inner to outer. Now, that's the same one that expands from the brain all the way to the spinal cord, or the spinal cord to the brain. It's only one. Now, on the subarachnoid space, so in the arachnoid, there's one below. In that subarachnoid space, which is this space right here, this space is filled with what? It's right there, CSF. So it circulates not only through the ventricular system, through central canal, but as well as into the what? Subarachnoid space. That's where you find your CSF in all these structures that we just mentioned. Whenever the CSF is being used, because as you use this fluid, the fluid contains right, sodium, potassium, chloride, glucose. So this fluid comes from where? From your blood plasma. From your blood plasma. There's no red blood cells. There's barely minimal or no protein at all in there. So we call this an ultrafiltrate of blood because it's really, really being filtered to really, really tiny molecules. Like what? Like the sodium, potassium, and the water. Just tiny ions. So whenever you use these ions within the brain, okay, whenever your brain uses or sends an electrical impulse, there's always going to be what? Waste products, like too much potassium, too much sodium, right? And that has to be in a constant circulation like a river. Imagine a river. So whenever you have excess of that CSF, how are you going to get it out of your brain and spinal cord? Because if you leave it there, excess amount of CSF, what's going to happen to your brain? You're going to compress your brain. Your skull is not going to expand because it's hard, correct? Your vertebral column is not going to expand because it's bone. So what's going to get compressed is the gelatinous structure, which is your brain and spinal cord. It's going to get compressed. So what's the escape? The escape is through the sinuses, dural sinuses. So what are those dural sinuses? Dural sinuses are not more than veins. Okay? So here are your dural folds. So within the dural folds, you have your superior sidal sinus and your inferior sidal sinus. Again, they're just venous system. They drain excess amount of CSF. Where to? Well, into the venous system of your body. Jugular vein, all the way into your heart. And then now becomes part of what? Your blood. Okay? Because remember we said it's a filtrate of the blood, correct? Now, what is the falx cerebri? It's a fibrous tissue which is part of your dura mater. When you actually took out the dura mater out of the brain when you were dissecting it, that dura mater had a little indentation in the middle, and that is known as the falx cerebri. So that one allows for a separation of both hemispheres, the right and the left. Where's the falx cerebri sits? It sits in the fissure. Remember the fissure? In that longitudinal fissure, that's where it sits. So when you remove that, you expose the longitudinal fissure. And then we have another one, which is known as Tentorium cereveli. If I remember this, I remember the word tent. Tent is like a roof, right? So it's the roof of your cerebellum. So hence the name Tentorium cereveli. Okay? So it divides your cerebrum from the cerebellum, or your cerebellum from the cerebrum. Okay? Now, how does that fluid circulate? And if you look at the fluid, what makes the CSF? What type of cells makes the CSF? I'm going to highlight choroid plexus, but what type of cells? Ependymal cells. When you have a group of ependymal cells, when you have a colony of ependymal cells, they make what we know as choroid plexus. Okay? So these ependymal cells are going to make your CSF. Your CSF is going to flow, and you can see, you can follow the arrows. And it's going to flow from the lateral to the third. From the third here, right, this is the third, to the fourth, this is the fourth. And then from there, it's going to go out laterally, all the way through here, on the subarynoid space. Or it's going to go to the central canal, all the way down onto L1 and L2. That is where your solid segment of your spinal cord ends. And it's going to go into your subarynoid space again. Does that make sense? And all that is going to circulate around your both your brain and your spinal cord. Okay? So what is the function of this cerebrospinal fluid? It cushions. It supports the brain because it gives them what? Nutrients, chemical messengers, and even takes out your waste products. Okay? So how do we get that X amount of CSF out of your subarynoid space into your sinuses? And that happens around here. Okay? What is around here? These are known as arachnoid granulations, like tiny granules. Okay? So the fluid goes into the granules, and the granules have bigger pores, and the fluid can actually dissipate into the sinuses. Now, the brain gets blood from the rest of the body, correct? And that blood, you have oxygen and nutrients. But the blood can actually be contaminated. How many of you have gotten sick? How many of you have gotten the flu? Right? Now, the question is, except you, how many of you have gotten an infection on the brain? Meningitis or encephalitis? No one. Why is that? Because you have something that protects you, and that is your blood-brain barrier. In other words, it's in the blood, between the blood vessels and your brain, and creates a barrier. What makes that happen? This type of cells, known as astrocytes. Osteocytes are in the bones. So, see, meningitis and encephalitis can happen because of a viral infection, can happen because of a bacterial infection, but it's highly uncommon. Why? Because of the astrocytes populations that you have. Now, the clinical application of this is, well, decrease the amount of infections, as well as tumors and masses. So, brain tumors, they tend to arise from either your meninges, right? Pia mater, arachnoid, and dura mater. But most of the tumors of the brain come from glia cells. And so the name is gliomas. So gliomas are the most common type of brain tumors. And what they do is they disrupt the brain barrier of the brain, and as well as when you give chemotherapy, that kills your astrocytes and disrupts the blood-brain barrier as well. Okay? And that's the treatment for it.